All right, grade 11. So this will be ended the last class when I was there last week. We looked at different parts of the alimentary canal and Subtopic 6.1 and the structure. You had to look under the microscope and recognize circular muscle, circular smooth muscle, and longitudinal smooth muscle, as well as the mucosa and epithelium. I asked you to look at this again in the lab on Monday. Hopefully you did. If you still aren't clear on these things, please make sure to come see me because these do tend to show up on IB exams, multiple choice questions, or even paper 2. So could be in your unit test, could be in a quiz. Make sure you prepare, okay? Okay, so let's talk about the pancreas. So the pancreas is important for hormone production. It produces both insulin and glycogen, which we'll talk about more in homeostasis. We talked about them last year in grade 10 if you were at RAS, which are important hormones for signaling about glucose and blood sugar, as well as enzymes. So lipase, amylase, and endopeptidase are all produced by the pancreas, which are 3 enzymes that you have to memorize. And the mix of all those things is called pancreatic fluid. Why are they put in the duodenum, those enzymes in pancreatic fluid? Why do they enter there and not? Why doesn't the chyme from the stomach go directly into the pancreas? Well, the pancreas is a gland. It's not a part of the alimentary canal, and it's meant to produce all those different structures. Remember, we looked at the pancreas in topic 1 as an example of the organelle structure, and we have all those exocrine cells in the pancreas that to produce all these hormones and enzymes. We also want to keep all the nutrients and everything in one place so we can have all the different enzymes and things coming into it to make it work efficiently in that one place. And then, of course, we have to memorize what those 3 enzymes digest. So lipase, amylase, and peptidase. What do they all work on, guys? Well, lipase is the easy one, works on lipids. Amylase works on carbohydrates, specifically starch. And endopeptidase works on proteins. So they're all going to be produced here and then entering the duodenum, the top of the small intestine, right up here. Okay. Same place the bile from the gallbladder is entering as well, which helps kind of break down the lipids. Doesn't digest them anyway, it just makes them breaks up the big chunks of lipids. So that way the so that way the enzymes can then work on them, because the lipids all clump together in the aqueous medium because they're hydrophobic. They're not going to be, the enzymes won't have access to them. So it does kind of separate them so that the enzymes can then work on it. So that way lipase can work on them. That's the pancreas. And then the small intestine. Look at the structure of the small intestine here again, where we have here is the tube itself with the layers of muscle, and then we have all these folds inside. And if we zoom in on one of these folds, you can see here we have the folds of the villus, and then it's covered in what are called microvilli. If we zoom in on a microvilli, villus, you can see we have a blood vessel here, capillary. We have lacteals, which are part of the lymph system we're not going to learn about in this course. We will talk about the blood system later on today. And then we have all these microvilli. These are to increase surface area. We want a really, really high surface area in the small intestine. Can you think of what function that might help support? Why is that important? Well, the big thing in small intestine is absorption, and we need to absorb things across membranes, and if I can increase my surface area, I'm increasing the membrane surface area, which means I'm going to have more absorption of nutrients and I'm going to waste less nutrients. That's both why the small intestine is so long, to give more time for absorption, and why we have these microvilli on the villi to increase surface area. It's not just a smooth tube, but it has all these protrusions on it. If you want to look at like a colonoscopy or an endoscopy video, you can kind of see the microvillus inside somebody. That's really kind of cool to see the structure of it. If you're easily grossed out, don't watch it. But if you're not, YouTube it. It's cool. Okay, so we're increasing surface area to increase absorption. And of course, the blood vessels inside, because we want the nutrients and minerals and vitamins to go into the bloodstream to be moved around the body where they're needed. Sugar to the different cells, vitamins and minerals are needed to make different structures and things in your body. Okay, so the key idea is the structure of the different components of the alimentary canal are designed to increase efficiency and efficacy in performing the function of each particular stage of digestion. So we have the pancreas to produce all of those enzymes and hormones. We have the small intestine with the folds and the villi and the microvilli to help increase absorption. We have the stomach that's got this thick mucosal lining that keeps being replaced because of stomach acid. We'll break down the lining. We have all these bits of it. We have the muscles in the esophagus. They're going to help move the food along through the bolus, through peristalsis. All these things are part of this idea. Okay, let's do membrane transport. So this is kind of back to topic 1, and because the end goal of digestion is to get the nutrients into your body and energy where it needs to be, it has to enter the bloodstream. So this movement can be with the concentration gradient or it can be against it. It really depends on the particular nutrient. So they cross membrane of the microvilli in the small intestine in different ways depending on if it's with the concentration gradient, it's going to be passive transport. So it could be just straight diffusion through the membrane or facilitated diffusion depending on the properties like the polarity and the size of the molecules. Remember back to topic 2, so reviewed membrane transport, or topic 1 membrane transport if you need to look at that. We also have active transport if we're going against the concentration gradient. So we have pumps and membranes, and we also have endocytosis to take things in. All those things work in your system to take nutrients, minerals, vitamins into the bloodstream from the system. Okay, so take a minute and look here at these different images of absorption across membranes and which is which type. So pause the video and figure it out. So this one is endocytosis, where we're making a vesicle and pulling these molecules inside the cell. I've got diffusion here, simple diffusion and facilitated diffusion, both, and apparently left the labels there, oops. And here I have active transport because I have ATP. Okay, key ideas. Transport is a key function of the digestive system, starting with food, the bolus, and then with smaller nutrient molecules. So this subtopic talks about digestion and absorption. So it's both part of it. A lot of the time when we think about digestion in everyday life, we forget about the absorption part. That's the whole point. If we just break things down and don't absorb them, we have done all this work with no benefit for our body. And the mechanism of transport depends on the properties of the molecules being transported and the structures through which they're being moved. So is it a polar molecule? Can it go through a amphipathic membrane layer? Again, go back to topic 1 to review that kind of membrane structure and transport stuff. All these topics come together. Okay, so last class on Monday, you did the diffusion osmosis bag of the dialysis tubing. Hopefully you guys got to do for that. We'll talk about it together when I come back. Here was a lab. Hopefully your stuff looks something like this. And you look at the muscle at the Alimentary Canal slides, some more of the genome part of the small intestine. Okay, so today's lesson is the blood system. So subtopic 6.2, there we go, is a long one. Lots of bits of this subtopic. We're going to talk about quite a bit of this as well when we do the heart dissection next class and review things, kind of connect all those things together. But there's a lot of also little kind of just facts to memorize with this subtopic. And they love to ask you what the sequence of blood flow for this subtopic on IB exam questions, the structure of the heart, all those kinds of things. We're going to talk about both of those when you do dissection next class. So there are 3 main blood vessels. Each has a different function, and because it's a different function, it's a different structure. So who can name them? They are the vein, the capillary, and the artery. So veins tend to be shown as blue in diagrams, arteries are red in diagrams, there's a transition in between. These colors are made up by scientists in diagrams, by biologists. They are not the actual colors of blood. Okay, your blood is not actually blue and red that way. You can look, your veins can look kind of bluish at the top, but the significant difference between the red and blue is not that way. It's exaggerated in diagrams to help us see the difference between oxygenated and deoxygenated blood. So arteries, they take blood away from the heart, and that blood is in a high pressure state because it's been pumped out by the heart, an incredibly muscular organ that's pumping the blood out. It's high pressure. Veins collect the blood from the body and bring it back to the heart, and now it's at low pressure for a long time since that blood was pumped by the heart. It's at much lower pressure than it was when it's first left the heart in an artery. And capillaries, they are very thin and they can be as thin as 1 cell thick. And so this is where we have gas exchange, nutrient exchange, anything coming in and out of the bloodstream happens at a capillary. So on your big muscle groups, your lungs, your intestine, you have a ton of capillary nets because they need to be able to absorb all the different nutrients, the different gases that you need to be able to function. Okay, so how does the structure of arteries help maintain pressure between pumps? So if we look at this diagram here I've got an artery, a vein, and capillary. Here's another diagram, the same thing. So we need to connect structure to function of this entire unit. So if I look at my artery here, it has a very thick wall and a very small opening. Okay, and the opening, what's going to be the opening? I will remember it. Anyway, so the thick wall here is muscular, can help withstand the pressure of the blood coming through. It's high pressure from the heart, and then we have a small opening to help maintain that pressure. It's a small area in the middle, so that we're not going to have a huge amount of space. So it compresses the blood in more smaller space equals higher pressure. It maintains that pressure to pump the blood throughout the entire body, because getting that oxygenated blood from the lungs through the heart to the whole body is a major priority for your body. And then the next thing is going to be capillaries. They're going to the capillary next. How thin are they? They're as thin as one cell. And this helps with diffusion because it's less area for the different nutrients, gases that are to pass through. And look at veins and their structures. Okay, how does the structure of veins help keep blood moving back to the heart despite low blood pressure? So they have a much thinner wall because the blood pressure is low, and you want to kind of get as much pressure as you can. And we have these little valves, these one-way valves. The blood's flowing this way, can pass the valve, no problem. But if it's trying to go through the valve and the heart, like between heartbeats, if let's say you're moving up your leg, if it passed through a one-way valve, it can't roll back down again between heartbeats. So between heartbeats, it kind of gets stuck at a one-way valve and it moves up a little further. This helps prevent the loss of backflow of blood because of gravity. So it keeps the whole kind of chain of blood in your blood vessels moving appropriately throughout the body. Okay, so here are 3 images. These are all blood vessels. And I want you to take a look and figure out which one is a vein, which one is an artery, which one is a capillary. The opening, the center of a blood vessel is called the lumen. The word came back to me, guys, I remember. So if you need help, here's a little diagram. So number 1 is the vein. It has the biggest lumen, the biggest opening, the thinnest walls. Number 2 is capillary. It's very, very small. And number 3 is, of course, the artery, the thick muscular walls and the smaller lumen. You can also see in this particular diagram, which blood vessel do you think that is? That is a vein, and I know it's a vein because there's a one-way valve in the center. Those little flaps, that's what a one-way valve looks like when it's not being held rigid because the cell is dead. They just cut a cross-section of the vein to get that slide. So identifying these in the microscope is an important skill for this unit. So make sure you can do this. You can put other images as well online and release exam papers. I can give you some more practice if you need it. Okay, here's our 4 key ideas from so far today. There are 3 types of blood vessels in the human body, each with a different function and a structure to match. Arteries have muscular walls and a narrow lumen to both sustain and maintain the high blood pressure of blood coming directly from the heart. Veins have narrow walls with one-way valves to help low-pressure blood make its way back to the heart. Capillaries have extremely thin walls to allow for exchange of gases at the lungs and nutrients in the intestines, as well as other materials throughout the entire system. So as always, when you see key idea slides, a moment to check your understanding. Make a note to yourself, oh, I get that, great, done, good, move on. I understand, but I have to memorize it. Or do you need help? You need to be reaching out to me to ask questions throughout the units, not just at the very end. Okay, so if you think you don't really get something, ask me to help you guys. I know this stuff, obviously. It's my job to help you. I'm happy to help you, so tell me if you have questions, okay? I love it when students ask questions. And right now, since I'm not physically there, you can send me an email and I can type you back an answer, or you can wait till I'm in class next time. Okay, so let's talk about William Harvey and his discovery. So it's another example of an old theory in biology that was refuted by science. So Harvey was one who refuted some ideas about anatomy, and he did work with blood circulation. Before Harvey, what they used to think was that your body constantly kind of used up blood. And then you had two types of blood. You have nutritive blood that comes from the liver, which carries like nutrients and energy. And then you have vital blood that comes to the heart that gives you like vital kind of life force, kind of like how we talked about vitalism in topic 2 about organic compounds being needed to have this vital force to make it. Same kind of idea. That was an ancient Greek idea. Obviously, it was very incorrect, but they just saw that people would lose blood when they're bleeding, and they'd get more. They wouldn't die. They'd eventually, if they didn't lose too much blood, they would get more blood back. So I think that's kind of the idea of it being used up came from. What Harvey did is did many, many dissections of a variety of different organisms, and what he found is the heart is a double pump, and the blood is circulated constantly. Now, without a microscope, without the kind of imaging technology we have today, without the way we have of preparing specimens that preserve the blood vessel, this was still pretty tricky for him to figure out. So his model here, this kind of diagram, is a little bit simple, like where he just has liver, heart, gut, not perfectly correct, but it's definitely a lot more correct than we thought before, the blood getting used up by the body, the nutritive blood and the vital blood idea. So, the four-chambered heart. The best way to think of our heart and any mammalian heart is it's two pumps. Okay, each pump has a certain destination, and each pump is built of two chambers. So each pump has a collection chamber, which is known as an atrium, or atria is the plural. We have the right and left atrium, and then we have the pumping chamber, which are the ventricles, the right and left ventricles. Okay, and the ventricles pump blood into an artery away from the heart. Now notice the right and left are opposite to where you're looking at them, right? And that's because these are anatomical directions. They're the directions based on the orientation of the organism. So if you were like lying on a table to be dissected, your side right and your side left is the reference they're going to use to talk about the directions, because the people doing dissecting or working on the lab or whatever it is, their positions can change moving around a specimen, but the specimen's orientation is constant. So use a specimen's orientation, we're talking about directions. So be careful with that with diagrams. If you're ever not sure, you're going to look at your own body and remember that the pointy part goes towards the center of your sternum, and that's going to be the right. Okay, so use your anatomy as an example. So there's four valves that control between those different chambers. So we have a valve here, and we have a valve going out up here, and a valve going out down here. Okay, so four valves. Here you have the tricuspid valve, the mitral valve, the aortic valve, and the pulmonary valve. So what I want you to do based on your notes and your knowledge so far, and what I just kind of told you with those different valves, write the scenes of blood flow through the heart and blood vessels. Write it on a whiteboard if you're not sure, but take a minute and write them out and try to kind of get those clear in your mind. We will go through it on the next slide, but pause the video, write them out yourself, kind of test your understanding from your homework reading. Okay, so hopefully you've written out your steps. We're going to go through that now and you can check your work. So this is my favorite diagram of the heart. Yes, I have a favorite diagram of the heart. This is it. So we're going to start with the vena cava. So we have the inferior vena cava and the superior vena cava taking in deoxygenated blood. Those are major veins, one from the top of the body, one from the bottom of the body, into the right atrium. Remember, the atria are the collecting chambers of the heart. So the blood's going to enter the right atrium, and it's deoxygenated. It's been throughout the whole body, come back to the heart to get pumped again, and move through the tricuspid valve into the right ventricle. Right ventricle, the ventricles are both pumping, so there's a pumping structure, pumping chamber, and when the heartbeat happens, it will pump the blood through the pulmonary valve, and pulmonary, of course, means lungs, poumon en français, okay, so pulmonary comes from Latin, and then goes to the pulmonary valve, the pulmonary artery. Remember, arteries always go away from the heart to the left lung and the right lung. The lungs give oxygen to the blood. We have gas exchange happening, which we'll talk about later on in this unit, how that works. And then it comes back to the heart through the pulmonary vein, where veins always go to the heart. And they're going to be entering the left atrium, because atria are collecting chambers, collecting that now oxygenated blood in the left atrium. And then the heart beats, the valve's going to open, the mitral valve will open, and that oxygenated blood will go into the left ventricle, which is one of the pumping chambers. It's the strongest chamber because it pumps the furthest. It's going to pump the blood through the aortic valve, through the aorta, which is the biggest artery in the body, and go over through the entire body to circulate again. So we look at this diagram. It doesn't have the detail of the heart, but it shows kind of the way it circuits around each other. So we have if you start with the deoxygenated blood coming in, same place, so throughout the whole body and through the lungs, you have the blood that was lacking in oxygen coming from the whole body, going through the superior and inferior vena cava into the right atrium, into the right ventricle, and then out to the lungs. So that's the pulmonary circuit. It's this path of going from the heart to the lungs, back to the heart again. It's going to get oxygen at the lungs and move back into the heart to the left side. The left atrium, the left ventricle, then it gets pumped out through the aorta, through the entire body. And then the body uses up that oxygen and moves it back to the heart again. So this is a systemic circuit from the heart to the body and back, and then in between that we have the pulmonary circuit from the heart to the lungs and back. Okay, so these are great diagrams to have. If you're doing digital notes, you can probably find these images pretty easily on Google image search. But make sure you have the steps, the sequence of this written down. Okay, it's really, really common question. Okay, so let's talk about how the valves are opening and closing to actually allow this to happen, and how you have the different contractions of different parts of the heart. Okay, so what we have here, if we start at the beginning, this table is a little bit confusing when you first look at it, but it really explains it really well. So we have systole and we have diastole. We have contractions, relaxing, opening, closing different valves. So all the different chambers are here and different kinds of valves are there. So at the beginning, kind of our first step, we have the atria are going to contract. Now they're not very muscular because they only have to contract the blood just down to the ventricle right below. It's not a big distance, it's following gravity, it's pretty easy to do. So when that happens, the AV valves, or the atrioventricular valves between the atrium and the ventricle, they're going to open, and then my semilunar valves, which go from the heart out the body, both through arteries, they're both going to be closed. We don't want the blood to flow from here directly out. We want to collect it in the ventricle. Okay, and then next is going to happen is now these valves that were just opened, okay, the atria are going to relax, the ventricles are going to contract, very muscular parts of the heart down here, and it's going to force these valves to close. And for a moment, the semilunar valves out of the heart toward the lungs and the whole body are going to also stay closed. This then forces the semilunar valves to open, which allows the blood to flow through up here through to the lungs, get oxygenated, to the pulmonary valve, and then back in, and then also through the aorta out from the left ventricle out to the entire body through the aorta. Once that is done, we want to give the blood a chance to come back in. Then all four valves are going to close. We're giving a chance for blood to flow into the atria from where they were before, for the ventricles to kind of be empty, and the valves open, blood flows into the ventricles, the atria, and it all begins again. So the specific sequence of blood pumping, you have two parts of your heartbeat, right? The bub, bub, bub. There are stethoscopes at the back of the room. If we don't get a chance to use them today, you can use them next class. I'll show you how to use them. Don't just take them and shove them in your ears. You can damage your eardrum, okay? You need someone to show you how to put them in properly, or you can hurt yourself. Okay, sorry. I had a student actually hurt themselves pretty bad playing around with stethoscopes one year. Didn't know what they were doing. Anyway, so your two parts of your heartbeat is you have the first part of your heartbeat is the atria contracting and the valves opening, and the second part, the ventricles contracting and the valves opening. So we have two parts. Really talented doctors love to practice, and nurses as well. They can actually hear your valves. And remind me to do this next class and see if anyone has a good enough hearing and it can be quiet enough to actually hear someone's valves opening and closing. It's really cool. Okay, so diastole is when you measure the blood pressure. So when we're moving blood through the heart, it's due to blood pressure is the main reason for this. And the change in pressure between the different chambers is what's going to open the different valves. So when a chamber is not contracting, we say it's in diastole, which means the pressure is going to be higher in the atrium than it is in the ventricles. Okay, and this is what keeps the valves, the cylinder valves, the tricuspid and mitral valves open, because the pressure is higher up here than it is down here. So that keeps this valve open. Then the much higher pressure in this valve here, okay, is going to keep the aortic pulmonary vein in the veins above, is going to keep these valves closed. So high pressure above keeps the valves below them closed. We have to have high pressure below to open those valves. So high pressure here and here means this valve and this valve are going to be open. And the high pressure here and here keeps these valves closed because they're much higher up. And the angle of the valve, look how these valves, the flaps go down and those flaps go up. Okay, that has to do with the way the pressure causes them to open or close. So in systole, that's what we call when the chamber is contracting. So when the heart muscles contract and the atria are contracting, so remember a smaller, thinner chambers for collecting blood, when they contract, there is pressure produced, but it's not very strong pressure because the walls are so very thin. Their big job is to collect the blood and then let it flow down to the ventricles. But that is enough pressure to kind of get the rest of the blood down to the ventricles and then close the valves, because most blood is already kind of moved down passively due to gravity during diastole. So then the ventricles are going to pump next, and the ventricles are really thick compared to the atria, very muscular. And when they undergo systole, remember systole is the contracting of the muscle, this really thick walls can produce a lot of pressure, and that's going to pump the blood all the way to the lungs in this case, and all in the entire body in this case. So this pressure difference is enough to then close the valves here, the tricuspid and mitral valves, the cylinder valves, the tricuspid and mitral valves, and then open up cylinder valves, also known as the pulmonary and aortic valves. As this pressure builds up and builds up, eventually the valves are going to open and the blood moves onto the lungs or onto the entire body. Okay, so here's your key ideas. The mammalian heart is a double pump constructed of 4 chambers, which facilitates a specific sequence of muscular pumps, pressure changes, and blood flow. Blood first enters the heart and is pumped to the lungs, to be oxygenated, then back to the heart and to the entire body. So the sequence of that flow is important as well. Remember again, key ideas, check just for understanding. Do you have any questions? Make a note if you have things you want to ask me either through email or when I'm back on campus. Okay, now how do we actually control the heart rate? I'm sure you've all noticed your heart rate changing in various situations, whether it's exercise or stress, excitement, any of those kinds of things. And it changes, your heart is beating constantly, thank goodness, and that's because cardiac muscle is very unique in the fact that it does not get tired. If you have healthy cardiac muscle, it's pretty much never going to get tired. And it's able to contract and relax on its own. That's called myasogenic muscle contractions. We don't have to tell our heart to beat. It does it automatically, and that's one of the things very unique to cardiac muscle. We do have microscopes that slides at cardiac muscle, so you can look and kind of compare them. What we need our nervous system for, the SA and AV nodes, is to pace your heartbeat. So we have a heartbeat that happens by itself, but the pacing of it to match our body's needs is done by the nervous system. So what happens is you have the SA node up here, the sinoatrial node, and it will get a signal from your brain that tells the heart, the atria to contract. So it tells this atrium and this atrium, they both contract. And then the AV node sends its signal to the SA node, so the SA node to the AV node after 0.1 of a second, and then the ventricles both contract. It's a pretty quick system of kind of regulating your heartbeat. If you've ever known anyone, maybe an older person, a grandparent, my grandpa had one who had a pacemaker, what basically happened is their nervous system and their heart is no longer functioning to regulate their heartbeat appropriately, so they put a little kind of computer chip in their body to help regulate the heartbeat. Now, the problem with this is it doesn't respond to signals from your brain, but it will give you a regular heartbeat all the time, which is better than an irregular heartbeat, of course. Okay, the other way we can control heart rate is when we have different CO2 levels. So the medulla part of your brain, it is able to sense an increase in carbon dioxide in your blood. And if you have too much CO2 in your blood, your heart rate will increase via the SA nodes. The medulla uses your nervous system, the SA node, to tell your body to increase the heart rate because you have too much CO2. Why is that a bad thing to have too much CO2? Well, it means you're not getting enough oxygen. Think about cellular respiration, about aerobic and anaerobic respiration. If we don't have enough oxygen, we have too much CO2, we're going to have to move into anaerobic respiration and produce lactic acid, which no one likes that feeling in their legs or where the muscles are, where we're producing lactate instead of pyruvate because we don't have enough oxygen. So your brain will regulate that. If you're exercising more and you need to have more oxygen, because you have too much CO2, your body will signal your heart to beat faster, pump the blood to the lungs faster, get that oxygen out to your body faster. You can also tell your SA node to fire more through chemical signals like hormones like epinephrine. So what other reason might you need to have a heart rate increase besides just high CO2 levels? Stress, fear, anxiety, excitement, all those kinds of things. We've evolved to adapt, and if we feel scared or threatened or anxious or something that require a lot of energy, we get hit with epinephrine, and then we end up having a higher heart rate. So we have more oxygen to our muscles, are ready to react more quickly to whatever the situation should require. So we're connecting evolution here, we're connecting topic 2 here, we're connecting all the different things. This entire course is interwoven. Okay, key ideas. While cardiac muscle can be independently, it relies on the nervous system to regulate its heart rate. The body responds to environmental changes to nervous and hormonal signals. One possible response is an increase in heart rate. So that's basic. You have to know about how the blood system functions, both the heart and the blood vessels. We're going to talk about what can go wrong now. So think about plaque and occlusions. So atherosclerosis is the fancy biological term, medical term, where we're talking about plaque buildup. So if you have plaque building up, this is an artery. We talk about the muscular wall, the relatively small lumen. We have buildup of plaque in the arteries. And at this point, it's becoming atherosclerosis. Why is this a bad thing? Why is this ideal and this really bad? Well, what's happening is we have a small occlusion where we have these builds of plaque in our blood vessels, which are fairly normal to happen, and then when it comes to atherosclerosis, we're actually restricting the blood flow very, very severely. Now, if this happens in a really, really critical artery, like cardiac artery feeding the heart, or one to your brain, or other really important parts of your body, it can cause huge impacts. Now, an occlusion is just where you have plaque buildup, and they do happen over the course of your life, depending on your diet, exercise, genetic factors, etc. But the severity of them, so atherosclerosis, we're actually blocking it almost completely. You can get surgery that kind of take almost like a pipe cleaner and clean out your blood vessels, but it's high risk, and they only do it when you really, really need it. So why would an occlusion to a coronary artery or another major blood vessel, why would that be much worse than a more peripheral blood vessel? Let's say I have an occlusion in an artery in my arm, my forearm feeding part of my hand. And then I have an artery feeding my brain or an artery going towards my heart, feeding the heart muscle itself. If it's in my hand, it would suck if my hand is affected and I don't have as much femoral control, but that's only going to affect one part of my body. If it's affecting my heart, then that's going to affect all the blood flow in my entire body, which affects absorption and transport of nutrients, affects gas exchange, affects communication in your body through hormones, all those things. So if it hits your brain, it can cause huge factors in your entire body. So depending where the occlusions are happening, they're more or less likely to do surgery to fix it. Okay, so key ideas. While plaque buildup in blood vessels is normal Too much plaque, sorry guys, can block blood vessels and block blood flow, leading to heart attack and other health conditions. Okay, that's the end of today's lesson. So for homework, you need to prepare for the heart dissection. Choose your partner and read the procedure being handed out to you right now on paper. This is going to be assessed. Your drawings, your measurements, and your analysis will all be assessed. Watch the video, and if you can't handle the video, watch the walkthrough. Check out the walkthrough on the website. You only get one heart. You can't redo it if you mess it up. If you cut something the wrong way, it's really hard to see things because it's like falling apart puzzle piece. Prepare, prepare, prepare. Okay, guys? I'll be here to help you, hopefully, but you really need to make sure that you are well prepared. We have some new dissection tools. Will help with making easier cuts and not damaging the heart as much, but yeah, be ready, okay? Any questions or comments, please feel free to email me. Hopefully back on campus soon.